Good afternoon, everybody. Welcome to the Kids First Program Spring Webinar 2023. We'll start now with a research showcase featuring the evolution of data in a collaborative landscape. Thank you for joining us today. Again, this is the Kids First Program Spring Webinar. I'm Marcia Fournier. I'm the program manager and I will be your host today. And my colleague, David Higgins from the Kids First Data Resource Center will be my co-host. I will start with an introduction and about the program and funding opportunities at NIH that support the Kids First program. And Dr. Higgins will introduce the research showcase with presentations by Dr. Seidman, Gillani, and Guy, and Heath. The Kids First is a common fund program administered by NICHD, along with NHLBI, NCI, and NHGRI. 10 years old, Gabriella Miller, a childhood brain cancer patient and advocate, inspired the program, authorized by Congress in 2014. Since 2015, the program has granted $12.6 million per year to develop a data resource for pediatric research. The scientific hypothesis of the program is the intersection between human development and cancer progression. Recent research has supported structural birth defects and childhood cancer share gene mutations. Also, multiple large studies have shown that structural birth defects increase risk of cancer during childhood and adulthood of affected individuals. The Kids First resources aim to allow sharing multiple data types across pediatric conditions, which enables a better view of a condition's underlying biology, speeding up the development of diagnostics, treatments, and prevention tools. The Kids First works through its Data Resource Center, genomic sequencing centers, and the NIH EXO-1 mechanism to support data generation and data sharing in the cloud. To date, the program has supported the public release of about 30 projects, 18 birth defects cohorts, and 15 cancer cohorts. Over 500 secondary users got granted data access, approved for secondary use of the data. And the program has made available nearly 30,000 genomes representing structural birth defects and childhood cancer patients and families. The Kids First is also collaborating and developing ways to cooperate with other data science resources, including the NIH Cloud Platform Interoperability Effort, and the NIH Common Fund Data Ecosystem and the dbGaP. Now I'm going to tell you a little bit how sausage is made. The Kids First, as I said, is empowered by the EXO-1 mechanism, which is an NIH support mechanism which will give access to selected projects to sequence cohorts. We allow the program tends to prioritize projects that allow broad data sharing, and these projects then receive access to the sequencing centers. And the sequencing centers, the Broad Institute and Hudson Alpha provide service to generate the genomic data from the DNA and RNA samples sent by our investigators. Once the data is completed, the Kids First Data Resource Center performs a key role on compiling this data, the genomic data with phenotypes and clinical data to deliver back to investigators. The data release to the public happened 6 months after the investigators received the data. So the investigators have 6 months exclusivity with the data before it's released to the public through the Data Resource Center portal and dbGaP. So more often than not, our investigators also, after receiving the data back, that may create new hypotheses for new cohorts being sequenced through new EXO-1s applications, but also create hypotheses for data analysis through other NIH mechanisms, and I will tell you about that in a minute. This cycle is a very collaborative cycle, so any investigators applying for the Kids First EXO-1 should be prepared to collaborate with the NIH staff, the Data Resource Center staff, and the sequencing centers. And through this collaborative effort, we can not only deliver back quality data to investigators, but share this data globally with researchers. Important things to keep in mind about the EXO-1 mechanism. Obviously, read the announcement in details. We expect a new announcement later this year for the 10th cycle of the Kids First EXO-1, so keep eyes on it. The EXO-1 gives access to generate genomic data that is performed in a designated Kids First sequencing centers, the Broad or Hudson Alpha. Also, keep in mind the EXO-1 does not fund the data analysis, even though we ask investigators to include the data analysis in their EXO-1 applications, but the data analysis can be funded through other NIH mechanisms such as RO3s or RO1s. The data with consent, we expect data with consent allowing broad sharing of the data. And the programs prioritize proposals that with inclusive and diverse cohorts with rich phenotypic and clinical data associated with the cohorts. I invite everybody to check projects that has been funded at the Common Fund Kids First page, and the URL is here, and I will share later after my presentation in the chat box as well. So feel free to share, to take a look on project that has been funded before to give you a sense of what was funded in the past. I would like to share also this graphic shows the body map with the different cohorts that are already in the public domain through the Data Resource Center and dbGaP. So today, this data is available not only for the original primary investigators, but now this data is available to globally to researchers. And over 500 users has been already approved for secondary use of this data. So I invite again you to check it out at the Data Resource Center portal or dbGaP, the Kids First that are available for researchers. And again, I would like to emphasize this data is not only available in the United States, but also available internationally through dbGaP. Now I'm going to tell you a little bit about funding opportunities in addition to the EXO-1 mechanism that I described to you that support data generation. The Kids First is supporting with different mechanisms, the data analysis. For instance, our Kids First Cloud Credit pilot is sponsored by the NIH, and the application is very simple through an email with the turnaround of 2 to 5 weeks. Send us an email. I will share again this URL through the chat after I finish my talk. And so feel free to take a look and send your applications. I will myself receive your emails for the cloud credits. The things that we ask when you're requesting cloud credits is that you pick a data set, Kids First data, and you can find that at the Kids First Data Resource portal, and also use Cavatica for your compute and analysis of the data. But all instructions are, you can refer to the Cloud Credit page, and you will see the description and questions you have to answer to send us your applications. Also, I want to mention RO3s that are specifically designed for Kids First data analysis, including the institutional sponsor RO3 for Kids First data analysis. This is open at the moment, and again, I will share that link to if you are interested, and also other RO3s that are relevant for the Kids First data, secondary analysis of the data, And these opportunities awards for up to 2 years with the budget for direct costs up to $100,000 a year. Preliminary data is not required. In addition to the RO3s that are relevant for the Kids First data analysis, obviously go ahead and do your search at the NIH grant portal to check it out, other opportunities that might be relevant to you. And I will finish my talk by sharing with you the Kids First vision of alleviating suffering from childhood cancer and the structural birth defects by fostering collaborative research to uncover the etiology of these diseases and supporting data sharing with the pediatric research community. With that, I will open for questions, and thank you again for attending the webinar today.
Great. Thank you so much, Marcia. On behalf of the Kids First Data Resource Center, I'm so happy to welcome everybody to our spring webinar, which we're calling the Research Showcase this year. To kind of introduce our 3 speakers that we have today, I want to give a little bit of an introduction to the Kids First Data Resource overall, and then how researchers are using our Kids First data. So what is the Kids First Data Resource? So if I can describe it very succinctly, I would say that the data resource is 28 harmonized genomic and clinical data sets to advance discoveries in pediatric cancer and structural birth defects. And so there's a lot there, as succinct as I could be. So let me try to break it down a little bit into some smaller pieces to really describe what Kids First and our data resource has to offer to investigators. So there's parts of this. So there's 28 data sets in pediatric cancer and structural birth defects. And so as Marcia described, a lot of Kids First is supported by the ECHO-X01 mechanism, where investigators can submit cohorts, which are then sequenced and then processed and ultimately released to dbGaP for secondary investigators around the world. So at this point, we have 28 different data sets that are available on our Kids First portal. And in fact, 2 of them are just launched this past month. So one, Kids First Charge Syndrome and Kids First Laterality Birth Defects were both our new data sets that just came out last month, and we continue to release more data month by month in the future. So if you want to learn more about these data sets, you can sign up to receive updates on these new releases and more, as we'll share here in the chat with you all if you're curious to learn more. So what for these data sets, what is available? So there's harmonized genomic data. And so for each of these participants or samples in the study, we call germline variants. We provide them in the GBCF format, which can then be combined together by investigators for a joint genotyping to allow kind of a rapid transition into the variant discovery phase. We also offer for our cancer studies, we offer a very robust somatic variant pipeline, which includes about 8 or 9 different variant callers of different types that call single nucleotide variants, copy number variants, and structural variants. So we have a very, very robust pipeline for that. For many of our birth defect studies, which are trio-based analyses, we offer joint called variants in that trio of mom, dad, and the proband child. Which allows and kind of accelerates the identification of de novo mutations in these birth defect research studies. So that's some of our genomic data, and then paired with that is our clinical data. So our phenotypes are mapped to the human phenotype ontology, or HPO, and our diagnoses are mapped to the Mondo disease ontology. And so these allow for investigators to be able to find the correct genomes, the correct participants that are relevant to their research questions, to make these discoveries. And everyone can search and build a cohort using these ontologies on our website, the Kids First Data Resource Portal. And so ultimately why we're doing this and why we're building this resource is that we want to advance discoveries for these kids. And so how are investigators using the Kids First Data Resource to advance discoveries? So one way that researchers can use Kids First data is that you can combine multiple Kids First data sets together. So there's many different Kids First data sets, and some of them have similar diagnoses or presentations. They can be combined together. So because these are harmonized, it really enables this very rapid combination. And so in fact, this is a little bit about what we're going to hear from our first speaker, Dr. Seidman, who's been combining Kids First data set to learn more about microtia risk in indigenous populations. Researchers can also use Kids First data to expand their current studies. And so if you have your own data set, you can add the Kids First data to it to get a larger set of samples, which increases your statistical power and allows you to be able to make new discoveries more easily. And we're going to hear a little bit more about this from our second speaker, Dr. Riaz Gillani, who investigates Ewing sarcoma. And then finally, researchers are bringing their own specialization to Kids First data. And so our Kids First data sets are, as we said, they're large, harmonized numbers of genomes that are all meant to be combined together. So if you have your own specialization, your own research question, you can use the Kids First data to test those hypotheses, test those tools, and make new discoveries in new fields as well. So this is our little introduction to our third speaker, Dr. Guy, who's going to talk to us about mitochondrial DNA in pediatric cancer. So with that, that's my setup for our three speakers today. And so I'm not going to take any more time because I want to get out of the way to hear these great talks that we have lined up. But again, if you have questions for any of our speakers, please put them in the chat, and then I'll try to facilitate them with our speakers as we go. So with that, I'm happy to introduce to everyone Dr. John Seidman. Dr. Seidman is the Henrietta B. and Frederick H. Berger Professor of Cardiovascular Genetics at the Harvard Medical School. He's going to talk to us about mapping genetic loci associated with microtia using Kids First whole genome data. So with that, I'm going to stop sharing my screen and pass it to Dr. Seidman.
So David and Marcia, thank you so much for that very nice introduction. I think it works very well with what I'd like to talk about today. So our group is actually associated with two different kinds of consortia that are involved in birth defects or structural birth anomalies. Congenital heart disease, which I'm not going to talk about today, there's a consortia, the bench to bassinet program that's run by the NHLBI, and that subfunds the PCGC. And in the course of that, the Gabriella program has sequenced a large number of trios. That's a father and a mother and an affected child. And that, as I say, I'm not going to have time to talk about that today. But what I would like to talk about today are our studies of microtia. So microtia can be diagnosed by almost anybody here because it's easily recognized as an abnormal or an unexpected change in the outer ear or the pinna. And the format of our study, for most of the individuals where possible, we collected blood and DNA from the father and the mother and from the affected child. And an important feature of both congenital heart diseases and microtia is that we very frequently see an affected child without seeing affected parents. So if it's genetic, then it means that the father or the mother carries a genetic defect, but they don't present with that defect. And it's an important question, I think, that the Gabriella Miller program will eventually help to explain, is how it is that you can have individuals who carry a variant which in their children causes a birth defect, but which in the parent is not apparent. But this format of using the parents and child was very powerful and was recognized not only by our group, but also by another group that was interested in craniofacial microsomia, and I'll tell you about that in a minute. But first, I wanted to just remind you of the features of microtia. So these are just here, you can see in these 5 pictures, a display of the different forms of microtia that can occur. exhibiting from a kind of an unusual pinna to one where it's completely absent. And it's these, in the cases where there's complete absence, which are most perhaps of concern to affected individuals, because that can lead to conductive hearing loss, and many of these individuals have significant hearing impairment in the ear with microtia. And it's quite striking as a developmental biologist to think that most microtia is unilateral. So I told you that this condition is already non-penetrant in that one of the parents can carry a variant that predisposes to disease, but not have any structural abnormality. Whereas in the child, only one of the two ears will actually be affected. So the other ear is again an example of non-penetrance. It's only 10% of the cases where microtia is bilateral. In general, we know that microtia is very rare. 1 to 5% of cases are familial. That is, there are other family members who are recognized to have microtia. We'll come back to that in a moment. And then, so as I said, microtia is extremely rare. Another important feature for both our study and for the other study funded by the Gabriella Miller program is that it's been recognized that in general, the incidence of microtia worldwide is quite low, about one microtia patient per 10,000 live births. But in Latin America, the frequency of microtia can go up can increase almost 15-fold, up to 17 per 10,000. And that actually provided the opportunity for us to do this study. We worked closely with both physicians, Ron Evie, who I don't believe is here today, and Usama Hamdan, who were at the time at the Massachusetts Eye and Ear Infirmary. And they actually were involved in medical missions that went to Latin America to do repair of pinna of individuals with microtia. And as part of those medical missions, several people from our group went and collected blood, and if possible, both parents and the child, but in multiple instances, just the affected individual, and consented them, of course, for genetic studies. And we've had that DNA available, and very kindly, the Gabriella Miller program agreed to sequence their whole genomes, and that provides the basis of what I'm going to talk to you about today. So we actually have used the data provided by the GMKF program to study two different kinds of disease. One was a very rare genetic program that was a single family that was present in the data set that they sequenced. And then more importantly, was to use the trios that I described to ask where is a single genetic variant present in their genomes that they all share, that it predisposes them to microtia. And again, we took advantage of two cohorts, one collected in our group from these medical missions, and the second was a cohort collected by Danielle Lucchetti and Carrie Heike, who recognized that there were orofacial cleft deficits in Latin American families. And it turns out we're both studying the same thing, but the clinical diagnosis was a little bit more broad in this Seattle group's study. But in the rare family that was present in our cohort, there were 5 individuals actually 4 that were indicated here by these arrows in a more recent birth. But the 4 whose whole genome sequences were determined by Gabriella Miller allowed us to say these 4 individuals shared one part of the genome, because using whole genome sequence, we could look for very rare sequences and ask where very rare variants. And the chances that each of these 4 individuals would share those rare variants was very small. And it allowed us and pointed us to a portion of chromosome 2, a 10 centimorgan region or so, that contained the FOXI3 gene, which is a transcription factor, which is known in dogs. There's a kind of a dog that doesn't have any ears, and it's because they lack FOXI3. So this was a very good candidate gene. And when we went back and looked at the genome DNA of the rest of the individuals in this family using an affected only method, it was very clear that a locus for microtia sits right in this region. And this was published earlier this year by Dan Quiet from our group and in collaboration with Andrew Timberlake. Actually, in the last couple of weeks, a second study has been published by Mao et al. With also finding that FOXI3 variants are very likely causes of microtia. It's quite I think, impressive that in all the world until these two studies came out, no one had ever seen this arginine 236 tryptophan variant. And just independently, our two groups found two different families, both of which share this missense mutation. So it's very likely that this missense mutation causes microtia in these families. But notice again that even though this is a monogenic trait, a single gene defect, a single amino acid change, the penetrance is actually remarkably low. Here, none of the affected individuals have an affected parent. So again, we have to understand, and these individuals are also unilateral. So there's lots of examples of non-penetrance, and it's an important question as to why or how these variants increase the risk of microtia without it being an absolute cause. So that was, those are rare. That explained like just one family and all of the subjects that we've collected collectively. So we wanted to understand more broadly, why is it that among individuals of Latin American ancestry, why is it that they had such a much higher risk for microtia? And to do that, we took advantage of a test called the transmission disequilibrium test. Actually, because these individuals are from relatively underrepresented minorities, their genomes, their sequences, their variants were not available on common arrays that are used for microarray analyses that are seen in people of European ancestry. And so that's certainly one reason why their analyses, this locus was not identified. But we're able to, with whole genome sequence and the sequence of the parents, we're able to do what's called the transmission disequilibrium test, where we take advantage of the fact that we can ask how likely is it that if an individual has a piece of DNA recognized here as an A, how likely is it that they transmit it to their affected child, or that they don't transmit it to their unaffected, or they don't transmit it. And so we looked through the entire genome for a place where a significant fraction of the 73 individuals who were probands, we had both parents of these 73, we had 73 trios. How likely was it that we would transmit that piece of DNA to their offspring? And we found a locus on chromosome 3 where there was what we call genome-wide significance. The chances that we would see this association by random chance is 1 in 10 to the 8th. It's bordering on one chance in 50 million, which is obviously quite rare and unexpected. And we can say that at this locus, if you inherited this piece of DNA, you were 5-fold more likely to develop microtia than other individuals. And we could replicate that with the second half of our study using 124 individuals where we didn't have their parents, and just did a straight up association study. The odds of that association by chance were 1 in 10 to the 7th or 4 in 10 to the 7th. And finally, using the second study funded by the GMKF, Danielle Luccheti's study, which had 68 trios, we found a third independent confirmation. So combined, the chances that this locus would show this association by random chance is approximately 1 in 10 to the 17th or 1 in 10 to the 18th. It's sort of like the chances that a single person is going to win the lottery twice. So that doesn't happen by chance. And it doesn't happen by chance that there's this piece of DNA that's inherited by all of these individuals who have microtia. So now a second question that we can start to ask is, how does this locus actually cause the condition? So the first way to answer that question is to ask, well, where does this piece of DNA sit on chromosome 3? What is it doing there? So the piece of DNA that's associated with microtia is located quite clearly between two genes that have been identified previously, ROBO1 and ROBO2. Just to remind the non-scientists in the audience. So this ROBO is a gene that's actually conserved all the way down to Drosophila of fruit flies. It was called roundabout in fruit flies. And you'll quickly say, well, of course, a fruit fly doesn't have an ear. So what is it doing with a ROBO1 gene or a ROBO2 gene? So a roundabout gene or a robo gene is involved in organogenesis. It brings together pieces of DNA and it creates structures. In Drosophila, the structures that we're talking about are not ears, of course, but other structures that are required for Drosophila function. But here we're saying this gene, this variant perturbs the expression of one of these genes, and we actually think it's ROBO1. An interesting question to molecular biologists is, here we have a segment of DNA that's about 50,000 base pairs downstream on the 3' end of ROBO1. So how on earth does it regulate expression? Well, there are two clues, and because we had these two lovely data sets from GMKF, we could actually map the location of the causal variant or the causal change in DNA quite precisely. And this landed right on top of a repeat sequence. But before I get there, I just wanted to point out one other important, interesting piece of biology. So we also were able to obtain the entire haplotype, all of the variants that are associated with this segment of DNA that are shared by all of these individuals who have microtia. And what we noted is that there were two variants that had been identified previously, rare variants that had been identified by in actually Chinese or Asian populations. So it turns out that there's a haplotype in Asia, which is associated not so much with microtia, but with a more broader classification, craniofacial microsomia. But the chances that these two haplotypes would be descended from one another is remarkably high. And so our current hypothesis is it's known that North America was populated, the current, what we think of as Amerindian populations, came from people who migrated across the Bering Straits. The first migration was 23,000 years ago. They migrated across the Bering Straits and then migrated first into North America and later all the way down into South America. And it appears that because there's an even higher enrichment in Latin America than there is in North America, we suspect that this variant was present in this very first migration. It's not clear that one of the scientific discussions is how many people actually walked across the Bering Straits 23,000 years ago. And it's somewhere, estimates range between a single small family of 10 or 15 nuclear individuals to as many as 1,000. In any event, in this migration, there was clearly an evolutionary bottleneck and an enrichment of the microtia variant, so that now in parts of Latin America, it's as I showed you, it's enriched 15-fold higher than expected in other parts of the world. And so we're saying that this is a very ancient haplotype that's goes way back to Asia. Many years ago. And then we can begin to ask, as I said, what's the mechanism? As I mentioned, we can map this variant quite precisely on chromosome 3, and it maps right to a repeat region. Repeat regions are of interest to geneticists because they can expand and contract during the course of, certainly in human evolution, but even somatically, they can expand and contract. And our working hypothesis, although we don't have data for this yet, is that perhaps the expansion, as I said, this haplotype is associated with a 5-fold increased risk, but there's a lot of non-penetrance. One possibility is that this region expands in those individuals who develop microtia, but doesn't expand in individuals who are protected. So it's just something to think, and it would have to expand somatically because an individual with microtia has a very small chance of transmitting that microtia onto their child. So it's a current hypothesis that this region might somehow be involved in modifying expression of ROBO1. A little further data that supports that hypothesis comes from a technique called high-C, or chromosome conformation capture, where we can ask if this region of DNA marked by this yellow box is interacting with any other portions of the ROBO2 or the ROBO1 locus. And these black lines point to regions of interaction. And you can see there's a specific interaction that was detected between this box, this region, this polymorphic region, and the promoter of ROBO1. So our current hypothesis is that changes in this polymorphic region in the 3' end of ROBO1 are altering ROBO1 expression during embryonic development. And it can happen in one ear and not the other, explaining in part the non-penetrance, the low penetrance that we see for this variant. So I'd like to stop there and just to remind you what I've told you today, that there seems to be a relatively common variant that's descended from an ancestral haplotype that arose in Asia and migrated across the Bering Straits into Latin America and has now become relatively prevalent. They're explaining the high prevalence of microtia among individuals of his Latin American descent, and that it's possible that this is related to a large repeat region present at the 3' end of ROBO1 that involves in changing ROBO1 expression. And an ongoing question, perhaps outside the scope of the Gabriella and Miller program, is to understand how changes in ROBO1 can alter ear structure and ear formation. And there's a number of interesting, useful animal models for beginning to think how we can figure out what Robo1 does during craniofacial development. And I'd like to stop there and to thank all of the people who did this work, in particular Dan Quiet, who's actually a pediatric cardiologist from Children's Hospital in Boston. We recruited him to come and work on congenital heart disease, but he's become such a bioinformaticist that he was able to think about this and congenital heart disease at the same time. And so we're very impressed with the work that he's put in here. The wet lab work was aided, in our group was aided by Josh Gorham. And the samples were collected by Barbara McDonough and other members of the lab, Yami Nicolau and Lourdes Dieck, who went to collect the samples during the medical missions. And we have a number of important collaborators, in particular Ron Evie, as I mentioned, is a ear, nose, and throat physician who specializes in treating patients with microtia. And we're very indebted to Melissa Tumblin and the Microtia Association for all of their help in providing new samples and new subjects as we needed them in our research, and Usam Hamdan, who organized the medical missions. And with that, I'll stop there and thank you very much.
So there is perhaps one thing you could comment on. So we have maybe some young investigators who are getting started with Kids First or joining us here. Do you have any wisdom that you could share on how to investigate rare disease, things that you might have picked up that you'd be willing to pass on?
Well, I mean, one is practical and one is that there are obviously new technologies coming online. I mean, one remarkable thing, obviously, is this short read. So just to remind everybody, Gabriella Miller has been focusing on short reads. And I think that's great. That was the technology that we should use. I hope that Gabriella Miller will transition to long reads. And so I feel like there's room for long read technology. And so if new investigators who are looking for kind of a new computational area, is to think about how computationally we can recognize these repeat sequences in the genome and then understand better how changing repeat lengths can alter gene expression. Because if you had asked me almost 15 minutes ago whether changing repeat lengths was going to change gene expression, I would have said, no, there's no chance. So, but it's clear that there are multiple examples where changing repeat lengths has a big effect, at least in congenital anomalies. So I feel like there's a whole field there.
So our next speaker is Dr. Riaz Gillani. Dr. Gillani is a pediatric oncologist and computational biologist who works at the Dana-Farber Cancer Institute at Boston Children's Hospital. So Dr. Gillani today, he's going to talk about unraveling germline risk factors associated with the development of Ewing sarcoma. And so with that, I will stop sharing and I'll pass it to Dr. Gillani.
Great. Thanks so much for the introduction, David. And thanks to the whole Kids First team for inviting me to speak today. So today I'll be talking a little bit about germline risk factors and the development of Ewing sarcoma. And a lot of the research I'm going to be sharing today has actually been enabled by the Ewing sarcoma Kids First study. And so just to kind of jump right in, these are my disclosures. And as far as providing a little bit of background in thinking about cancer predisposition, specifically as it pertains to pediatric cancers, there have been a number of studies that have come out over the last decade or so that have suggested that about 10% of pediatric cancers are attributable to germline risk factors. These studies, while seminal, have also been kind of very focused in how they've defined this figure. And so they're often looking at a set of kind of known cancer predisposition genes and looking at either rare variants or common variants. But as data sets grow, numbers grow, and we broaden how and where we look, we're bound to find more. And another framework, just kind of building off of some of the concepts that Dr. Seidman spoke to, is the rare common variant framework, which is helpful for thinking about germline risk. And so this is illustrated in this plot over here. Rare variants, by definition, are less frequent in the population, but often have a much higher impact on overall disease risk, whereas common variants are relatively more common by definition, but have a lower impact on risk. And in Ewing sarcoma, a lot of prior work has centered on common population variants that have been identified as susceptibility loci from GWAS studies. There was actually a great paper that came out a few years ago that had focused more on the rare variant angle and had identified risk factors in DNA damage repair genes. And this opened up a whole host of questions, and specifically kind of thinking about what are these variants doing? Are they happening more than one would expect by chance if we looked at patients who didn't have Ewing sarcoma? And so we set out to explore this further within the context of Ewing sarcoma, enabled by the Gabriella Miller Kits first study, as well as other large consortia data. For context, the need for a better understanding of germline predisposition is particularly great in Ewing sarcoma, which is the second most common bone tumor in children and adolescents in the US. And a better understanding of germline predisposition can not only enable us to improve how we screen for risk, but also understand central biology so that we can better inform how we approach and treat these patients, which as of right now, with the exception of some variations here or there, are largely treated very similarly. And so the objective in our work was to evaluate differential enrichment of rare pathogenic variants in Ewing sarcoma, and we sought to contextualize this through comparisons with other pediatric sarcoma subtypes, specifically rhabdomyosarcoma and osteosarcoma. And we also wanted to understand inheritance of these risk factors as well. And so to approach this question, we leveraged data from Gabriella Miller Kits first, St. Jude Cloud, and other studies to put together two cohorts, essentially. The first of which was a pan-cancer cohort comprised of osteosarcoma, rhabdomyosarcoma, and Ewing sarcoma patients, as well as other sarcoma patients with other sarcoma subtypes, matched with cancer-free controls. And then in our validation cohort, we honed in on Ewing sarcoma, and this is where the Gabriella Miller Kits first data set came in particularly handy, because in addition to looking at these in a case control fashion, we also looked at these cases in the context of trios that were available. And the focus of our study was on bona fide cancer predisposition genes, so a set of about 140 genes that have been identified in the literature and other contexts to be important in the predisposition to various adult and pediatric cancers. And so across the discovery and validation cohorts, our populations were largely pediatric, as to be expected, and we had a roughly even split between sexes. And we started by inferring ancestry using common population variants and principal component analysis, such that we could be confident that in comparing cases and controls, we were controlling for some of the kind of most common confounding variables which are related to ancestry. And in doing so, what we saw was that most of our patients, but not all, were of European ancestry, but we had a good representation from other populations as well. The data I'll be sharing today is focused on the European ancestry. We carried out analysis on European and all ancestries, but for power considerations, we honed in on the European subset of our population. And so getting to some results, what we saw was that across our entire pan-sarcoma cohort, not surprisingly, germline variants in TP53 were enriched in patients with sarcoma generally versus cancer-free controls. We saw some signals that have been reported before in PTPN11 and DICER1, and then we saw a modest signal in FANC-C, a Fanconi anemia gene. And diving a little bit deeper into specific histologies, we started to see different patterns emerging when we compared Ewing sarcoma with osteosarcoma and rhabdomyosarcoma. And so in osteosarcoma, we were able to recover previously reported signals in TP53, RECQL4, RB1. In rhabdomyosarcoma, again, a signal in TP53, BRCA2 and DICER1 variants have also been reported. And then in Ewing sarcoma, the landscape was overall silent with the exception of 3 rare pathogenic variants that drove this nominal enrichment in FANC-C relative to cancer-free controls. And in turning to our validation cohort, we again saw this signal, again driven by 3 separate cases harboring pathogenic FANC-C variants relative to cancer-free controls. And these were monoallelic variants, heterozygous variants, which in contrast to patients who actually have Fanconi anemia, these variants were only present in the heterozygous state. It was kind of curious because it's different than the paradigm that we normally think about these variants operating to increase cancer risk in. And in our validation cohort, where the signal was more modest, we carried out a sensitivity analysis outlined on the right to demonstrate what we might see if we were to see a higher rate of variance in the population, depending on whether there was external validity with the match controls that we had ended up using. And we found that over a broad range of baseline population frequencies, this signal seemed to persist. And zooming out, again, using the Gabriella Middle Kit's first data, we saw accompanying signals in CHEK2 and FANC-A, which are DNA damage repair genes, and then borderline signals in ERCC4 and NBN, other DNA damage repair genes. And interestingly, this was accompanied by, as you can see on the graph on the right, a relative depletion or absence of germline pathogenic TP53 variants in Ewing sarcoma, which stood in contrast with rhabdomyosarcoma, osteosarcoma, and all sarcoma subtypes. And so we were curious about these FANC-C variants, but then also these broader DNA damage repair variants that we were identifying and how they may be operating to increase risk. And what we found was that in all cases where a DNA damage repair risk variant was present in one of these genes, it was concurrently present in probands, so patients afflicted with Ewing sarcoma, but parents as well, suggesting that these were again, kind of going along with some prior themes Dr. Seidman touched upon, moderate penetrance variants that seem to be increasing risk, but not in and of themselves a complete explanation for disease risk. And so our conclusions from this work so far have been that this recurrent enrichment of heterozygous pathogenic germline variants in FANC-C provides evidence for its role in increasing risk for some individuals with Ewing sarcoma. But what's particularly interesting to us is what this may kind of represent more broadly in terms of risk for Ewing sarcoma outside of patients who have germline pathogenic FANC-C variants. And specifically, Ewing sarcoma is a almost universally translocation-driven cancer. And so there's a pretty classic genomic rearrangement that happens in the tumors of these patients. And so an explanation as to why that rearrangement happens has been elusive. And we think this points us in a very interesting direction in terms of exploring that link between these germline risk factors and this tumor translocation further. As I mentioned, these variants seem to have modest effect sizes and are likely acting in concert with a complex, either polygenic or environmental risk milieu. And they're universally inherited as well, as opposed to arising de novo. And so that last point we thought was a particular boon in terms of thinking about how to explore other types of variants, again, focusing on rare variants that may be increasing risk. And specifically, we have started to turn to germline structural variants. And so the field has done a lot of work in short nucleotide variants and insertions and deletions that can be kind of captured by more traditional variant calling methods, but the area of germline structural variation is relatively underexplored. And if we can use the observation of risk variants usually being autosomally inherited in our patients, it gives us an opportunity to validate what we're seeing in our probands in parents as well. And that's particularly important for structural variants, which can be a little bit harder to validate. And the field of population genetics has actually done a great job in developing a robust framework to dissect the contribution of germline structural variants further. And this paper, led by Ryan Collins, Harrison Brand, and Michael Talkowski, outlined the use of a tool called GAT-KSV to enable an evaluation of germline structural variation in patients who don't have cancer. And so we sought to apply this framework to our patients, and we started to do this using our trio data sets from Gabriella Miller Kids. And what we're seeing is that we are finding variants that were previously missed by short variant calling methods that seem to be impacting genes that could give us a fuller perspective on risk factors for Ewing sarcoma. And so what's shown here are IGV screenshots with a coverage track of an affected patient with Ewing sarcoma at the top, and then one parent in the middle, and then the other parent at the bottom. And any dips or increases in coverage would reflect either a deletion or a duplication. And it can be sometimes difficult to differentiate an artifact from a true deletion or a duplication. And so this is where the parents actually come in very handy to validate what we're seeing is real. And so we can see here that in this case on the left, there is an inherited deletion impacting an exon in FANC-I, a Fanconi anemia gene that's seen in the proband and also one of the parents highlighted here in the center. And then over here on the right, again, we see a deletion. This is a larger deletion spanning the entire FANC-A locus. It's FANC-A is another Fanconi anemia gene, and this is seen in the proband as well as one of the parents. And so we're actually interested in exploring this further and kind of more comprehensively. And so to do this, we'll be building off the rich data set from Gabriella Miller Kids and coupling it with other data sets, including cancer-free control data sets from 1,000 genomes from TopMed, and then other Ewing sarcoma cases from the literature to more comprehensively evaluate the germline structural variant landscape in Ewing sarcoma. And so with that, I'd just like to say thank you to Gabriella Miller Kids for enabling a lot of this work. Thank you to my mentors, Ellie Van Allen, Katie Janeway, and Brian Crompton, my collaborators, specifically Ryan Collins, who's really been moving forward a lot of the work with germline structural variants that I mentioned at the end, and then Saud and Sunghan and others who've really enabled a lot of the work around short variants that I shared earlier, advisors, and then my funding sources. Thanks so much again.
Great. Thank you so much, Dr. Gillani. We have a question from one of our other panelists. I think Dr. Seidman wanted to ask a question, so I'll pass the microphone to him.
Perfect.
So Riaz, that was a great talk.
Thank you.
Just to clarify, what's the frequency of Ewing sarcoma in the general population?
Yeah, it's incredibly rare. We're talking about kind of on the order of 1 to 5 in 1 million. And so I think that works out to be about 200 to 300 cases annually per year in the US. But that's about the order of magnitude we're looking at. So
two things actually. One, so PTPN11, I was really struck by that because that's the leading cause of unexplained congenital heart disease. And so PTPN11 mutations also cause Noonan syndrome, and that's a very common. They're associated with congenital heart disease. I'm just curious, have you looked for the overlap between the mutations that you see in Ewing sarcoma and the variants that are seen in PTPN11, which I believe are dominant, was basically poison polypeptide variants, but I don't know if there's any overlap.
So just to clarify, the PTPN11 signal was actually in the pan sarcoma cohort, and so it's actually driven by mostly non-Ewing's cases, actually. So it was other sarcoma subtypes, soft tissue sarcomas. On the point of PTPN11, though, it's interesting that that kind of caught your eye, because not only is it a signal in this kind of broader sarcoma cohort, but it's also a signal that we see in other pediatric cancer subtypes, including neuroblastoma, which is a relatively common extracranial solid tumor. And then, you mentioned the link with congenital heart disease. So, PTPN11 is obviously kind of associated with Noonan syndrome. And so I think just going back to like the broader theme of the connections between these different diseases that we all care about, there are these common themes that seem to emerge with regards to linking genetic etiologies. So I know that doesn't directly answer your question, but I just wanted to clarify that the PTPN11 signal wasn't Ewing sarcoma specific.
Thank you.
Yep.
I want to take advantage that Dr. Seidman and Gillani is here together and ask you both how difficult was for you, for Dr. Seidman, to combine Kids First data sets, and Dr. Gillani combining Kids First data sets with your own data. This is a question we get from researchers, and I think it would be great if you can expand on that, like more technical aspect of your research.
Sure. I think that there are considerations like batch effects that you have to take into account when you're pulling data from different places in terms of how things were processed and how variants are called. And so what we chose to do in our study is we start at the level of raw sequencing data and then called variants all the same from different studies. And so, I think that it wasn't difficult in the sense that, it could be done and the data was there and it was accessible. And I think sometimes the hardest thing is getting 100 gigabase file onto, your cloud computing platform. That being said, I think the harmonized data, both when it comes to germline questions, but then also when it comes to more tumor and cancer focused questions, is actually a really great asset, because there's a lot of opportunity, I think, where it's less important to kind of think about those upstream technical factors, where you can actually combine data sets relatively easily using those downstream harmonized data sets that Kids First makes available. And so I think it can be done at different levels depending on the question that you're trying to ask, but you just have to think about confounders and what level it makes sense to combine data at.
Yeah. So we just, in fairness, the microtia cohort was GMKF1. It was like one of your very, very early cohorts. And then the congenital heart disease samples have been collected over multiple programs. So they're in different tranches. So microtia, to be fair, I think the harmonization early on probably wasn't all it might be. So we, as Riaz said, we went back to basically the GVCFs that you all make are great, and we're able to use those and we can apply them to any variant calling package that we want. So we found that the GVCFs were perhaps the most useful package going forward with congenital heart disease, just in part because we've been collecting them over years. We tend to still go back to the GVCFs. I do think we all need to work on structural variants. The structural variant calling packages are still getting better. And but in general, technically putting the data together worked really, really well. It's great that they're all called on the same platform, and sequences that are done in one center versus another makes no difference whatsoever. It's way more, in fact, I was going to ask Riaz, it's way more of an issue of sort of selection bias. So when you see many more European subjects, is that because there are more Ewing sarcoma among individuals of European descent, or rather your centers are collecting samples that have a much bigger base of European ancestry patients?
I think the latter is an issue in pediatric cancer research more broadly. Specifically when it comes to Ewing sarcoma, though, Ewing sarcoma does have a preponderance in patients of European ancestry. So I suspect here that what was sampled is actually pretty representative of what we would expect to see in terms of in terms of the population. And so, if you were to look across thousands and thousands of cases, then I think you would eventually get to the point where you have reasonable representation of other ancestries. But Ewing sarcoma just so happens to happen more in patients of European ancestry.
That's a little surprising. To be honest, that's a little surprising when they're rare variants. So, I mean, super rare variants have presumably reflected de novo event. Yes, there are more kind of de novo events among older chromosomes like those of African ancestry, but it's not all that dramatic. I mean, the distribution you showed was really quite surprising. And it's hard to imagine that there's some reason why rare variants would occur in FANC1 more in Europeans than in people of other ancestries. But yeah.
Yeah. And I think it's definitely a story that we want to kind of develop further and explore further. I think that as far as kind of thinking about the FANCC variants, I think that I've wondered this is I don't know if there's if it's not FANC-C, is it something else, right? In these other cases that behaves like FANC-C, and so I definitely think we need to kind of flesh that out further.
That's great.
Great. I do want to keep it.
Answer your question.
Great comments, and also I want to thank you for your comment about long reads. We expect to in the near future to release data from our long read pilot, and hope this will inform the program about how to move forward, as this is a area of fast development. So thank you for that comment and for your comments on the use of the Kids First data. Let David move as we are good on time. I don't want to.
Yeah, we're doing good. I want to maybe one quick last question from the Q&A. So there's an attendee is asking for Dr. Gillani, do you have any ideas about why the confidence intervals of the odd ratios were so large in the rhabdomyosarcoma cohort compared to the other cohorts?
Yeah, no, thanks for that question. I mean, I think that there's a few places. I think rhabdo maybe was one of them, but then there were other areas as well. I think the confidence intervals were probably the most narrow for TP53. But in total, we are looking at rare variants, which we kind of defined as less than a 1% frequency in the population as a baseline filter. And so I think with rarer and rarer events, the confidence intervals were just larger. And so I think some of the rare events were Dicer1 and SDHD, and I just went back and looked in rhabdo, and I think that's what drives the while the point estimate is to the right, the confidence interval is very broad.
Great, thank you so much. So our third researcher going to present today is Dr. Xiaowu Guy. Dr. Guy is professor of clinical pathology and the director of bioinformatics at the Center for Personalized Medicine at Children's Hospital of Los Angeles. And Dr. Guy is going to be talking to us today about mitochondrial DNA mutation in pediatric cancer.
Great, and I thank the Kids First team for building such a tremendous resource and also for the invitation to tell you our story and trying to understand the mitochondrial DNA mutations in pediatric cancers. The story consists of three parts. One is trying to define the landscape of mitochondrial DNA mutations in pediatric malignancies. The second is trying to take a focused look of mitochondrial DNA mutations in pediatric CNS tumors. And the third is trying to understand the contributions of mitochondrial genetics to the susceptibility of Ewing sarcoma, which actually you just heard from Dr. Gillani. So start with the landscape study. In this study, actually, we used the whole genome sequencing data set of 621 pediatric cancer patients that fall into three major categories: hematopoietic malignancies, solid tumor, and brain tumors. So what we found in total 391 mitochondrial DNA mutations scattered across the entire circular mitochondrial DNA genome shown on the left. As you can see that these mitochondrial DNA mutations belong to 6 different groups: synonymous, D-loop, loss of function, missense, and the RNA mutation and TR mutation colored accordingly. And they also occur at different heteroplasmy levels depicted by these concentric circles, with the innermost circle being the lowest heteroplasmy and the outermost circle being the highest heteroplasmy, or roughly 100% heteroplasmy, homoplasmy. And immediately, if you just look at this circle, this plot long enough, you see that there's some patterns, right? So a lot more missense mutations compared to synonymous mutations. In fact, there are a lot more loss of function mutations, right? And compared to synonymous mutations as well. And they tend to cluster. They seem to be several hotspots of loss of function mutations in COX3 and ND4 and the ND5 genes as well. So these are all very interesting patterns that we observed. We have tumor samples from all 23 different subtypes, and we did some categorical analysis to show that in this figure, panel A, that there's an average number of mitochondrial DNA mutations per sample varies significantly from one subtype to another subtype, with the lowest observed in patients of low-grade glioma and the highest in adrenocortical carcinoma. And similarly, in terms of mitochondrial heteroplasmy, you see that it's also lowest observed in the patients of low-grade glioma, but very high in patients of Ewing sarcoma, and also some couple other tumors as well. In terms of this particular class of mutations that we're most interested, the loss of function mutations, they are again really are never seen, in fact, in patients of low-grade glioma, but commonly seen in some other tumor types like Ewing sarcoma, adrenocortical carcinoma, and so on. And putting things all together, you see that look at the composition of the mitochondrial DNA mutations, they vary significantly from one subtype of the tumor to another subtype of tumor, as shown in panel D of this figure. So you can also group the patients by the mitochondrial haplogroup that they belong to, right? And so we look at this mitochondrial group of these patients, we see that two group of patients, in fact, stand out. The group of patients with mitochondrial haplogroup H, the mostly European ancestry, and the mitochondrial haplogroup L, which is mostly African ancestry. You see that there's significant difference. The patients of H haplogroup tend to have a higher number of mitochondrial DNA mutations in the tumor than the patients of haplogroup L, and this is actually statistically significant. Looking at the composition of these mitochondrial DNA mutations, you also see the difference between the tumors of patients from these two different haplogroup H versus L. In L, in fact, you don't see surprisingly any mutations that are synonymous or tRNA and RNA mutations. So looking at the germline variants, we found it's very interesting that there are 4 patients having a homoplasmic mitochondrial DNA variant in both the tumor and matched normal samples that are known to cause primary mitochondrial diseases. And the 3 more patients having heteroplasmic mitochondrial DNA variant that are known to cause mitochondrial diseases as well. So altogether, 7 patients. There's 7 out of 621 patients, or a little bit over 1% of the patients having pathogenic mitochondrial DNA variants. And that's actually higher than the prevalence rate of mitochondrial diseases, right? This suggests to us that there's a potential increased risk associated with people carrying these primary mitochondrial diseases, or increased risk for pediatric cancers. And look at the profile of mitochondrial DNA mutations observed in the pediatric cancers compared to what we found published studies in the adult cancers. You see that the profiles are largely similar, right? There is only major difference seen is heteroplasmy level. Most of the mitochondrial DNA variants observed in the pediatric cancers are at low heteroplasmy, but in adult cancer, they tend to occur on higher heteroplasmy. But look at the pattern is very similar. You see a lot more missense mutation compared to synonymous mutations. And there are quite a number of loss of function mutations as well. This is actually quite different from the pattern you observe from matched normal tissue of these patients, as well as from tissues of non-cancer patients that we tested at our center. You see that there are, in fact, essentially none of loss of function mutation identified in any of these samples that we examined. And there are actually in the fewer, in fact, and the missense mutation compared to actually the synonymous mutations as well. Now, this ratio is quite interesting. We call DNA-DS ratio. This is a ratio of non-synonymous variant comparing to the number of synonymous variants. And this blue distribution represents what you expect from a random permutation, the ratio, DNA-DS ratio, which is roughly 2:1 in the normal tissues. But the dashed line on the left showing the observed DNA-DS ratio that we found in all these samples, and which is actually significantly lower than 1:1, and it's roughly 0.5:1. That shows there's some sort of purifying effect happening in the normal cells for getting rid of the deleterious or non-synonymous mitochondrial DNA variants. The opposite, in fact, is true in the tumor cells. You see that the pink distribution represents what you expect in terms of DNA-DS ratio seen in the tumor cells from a random permutation analysis. This ratio is roughly 3:1, and the dashed line on the right-hand side showing you the observed DNA-DS ratio we saw in all this data set, which is almost close to 5:1. That means there's some sort of selection for the deleterious or non-synonymous mitochondrial DNA variants in the tumor cells. And look at these 45 loss of function variants that were most interested. You see that they're mostly occurring in the complex 1 genes, especially in these 2 genes highlighted in red, ND5 and ND4. Looking from another angle, you see that of course, they occur in all different types of pediatric cancers, but they mostly often occur in the solid tumors, adenocarcinoma, osteosarcoma, and Ewing sarcoma. They all have a number of loss of function mutations in the mitochondrial DNA genome as we identified. So these mitochondrial DNA mutations, loss of function mutations, do not occur by random chance all over the place, right? So there seem to be some patterns. There seem to be some hotspots. So from permutation analysis, we found indeed there are, in fact, 4 hotspots represented by these 4 red bars shown in the middle. The height of the bar represents the likelihood of this being from, in fact, pure random chance. This is actually equal to minus log 10 p-value, p being the probability of this from random chance. The higher the bar, the less likely, of course, this being from random occurrence. So you can see that there are 2 hotspots for loss of function mutation in ND4, 1 hotspot in ND5, and another hotspot in COX3 gene as well. Similarly, there's also hotspot of TRA mutations in between ND2 and the ND1 genes as well, and as we identify from the permutation analysis. Now, so this actually should give you essentially bird's eye view of mitochondrial mutations occurring in pediatric cancer. You know that they occur quite often, they occur differently in different tumor types, and they may have a different degree of contribution to different tumor types. So in this study, we take a focused look at the pediatric CNS tumors by using this the CBTDC dataset made available by Kids First resource. So we mined the 545 tumor normal paired whole genome sequencing data set from Kids First, representing this number of CNS tumor subtypes shown on the right-hand side with the bar chart, pie chart. See that we found that this number of mitochondrial mutations from all different classes shown at the bottom of this figure, and from all these different subtypes, CNS subtypes shown at the top of this figure. You see that again, like there are a large number of missense mutations, frame shifting mutations, stop gain mutations. They all actually they can be discovered from all different subtypes of the CNS tumors shown at the top. So they are very interesting and consistent with what we observed early on from the pan-cancer study. Looking at the different subtypes, we see that the subtypes, in fact, have a significantly lower or higher number of mitochondrial DNA mutations. In this comparison, we compare low-grade glioma versus high-grade glioma. They have different grade. You see that the low-grade glioma has a significant fewer mitochondrial mutation than the high-grade glioma, which is very interesting to us. And this is also true with heteroplasmy level for mitochondrial mutation. It differs from one subtype to another subtype. Consistent with the pan-cancer study, we observe a number of loss of function mutations in the complex 1 genes, especially in the ND5 and the ND4 genes. Although looking at the mitochondrial haplogroups in these patients, we did not observe any significant enrichment or impoverishment of any mitochondrial haplogroup in any of the subtypes of CNS tumors. And look at the profile of mitochondrial mutations across all these CNS tumor subtypes, you see that the pattern, in fact, is very similar to what we observed from the pan-cancer study. The only difference, of course, is color was color schema was used differently in this study, and there are a lot more missense mutations comparing to the synonymous mutations. There are quite a few, again, like frame shifting mutations, but they tend to occur at lower cytoplasmy compared to that the ones observed in the adult cancers. And in the study, we found a number of patients, in fact, with multiple tumor types, and that makes these patients quite interesting. In this example, we have a patient with gliofibroma and with a primary tumor shown on the very left, and the first relapse shown in the middle, and the second relapse shown on the right-hand side. You see that this patient is quite interesting and is found to have one mitochondrial variant observed at 50% heteroplasmy, pointed out by this red arrow, and then found in the first relapse at 75% heteroplasmy, then at the second relapse at 100% heteroplasmy. So that's indicative that there's some sort of clonal expansion, and then, of course, this lineage of tumor cell become the dominant, eventually the exclusive lineage of tumor cells in the second relapse. And in the second relapse, you see the emergence of loss of function mutations in ND2 gene as well, suggestive this mutation may be contributing to the tumor progression. So that is about the CNS tumors. You know that mitochondrial mutation is very interesting and very important for CNS tumors as well. And so you probably heard me mentioning the Ewing sarcoma several times already, right? You heard from last talk and from Dr. Gillani that there's strong bias for patients with from European ancestry. So in our previous study, we found that mitochondria may have something to do with Ewing sarcoma as well. So you see that there are higher number of mitochondrial mutations in the Ewing sarcoma, and the heteroplasmy level tend to be higher in Ewing sarcoma as well. And there's one patient with Ewing sarcoma patient carrying, in fact, a pathogenic mitochondrial DNA variant, and the mitochondrial loss of function mutations also more frequently occur in Ewing sarcoma as well compared to other tumor subtypes. So that makes it very interesting for us to ask the question, what is the contribution of mitochondrial genetics to the susceptibility of Ewing sarcoma? Again, in this case, we take advantage of the tremendous resource provided by Kids First, and we mine this Ewing sarcoma cohort. And we look at it from mitochondrial angle, right? So we first of all, we determine the mitochondrial haplogroup using the Farmer tube that we developed in the past. And showing these diagrams that indeed, actually, most of the patients, Ewing sarcoma patients, in fact, have a mitochondrial group, mitochondrial haplogroup belonging to this R micro haplogroup. In fact, 375 of them out of a total of 420 Ewing sarcoma patients belong to the R micro haplogroup. There's significant enrichment of this European haplogroup in this patient. And on the other hand, there's a significant impoverishment of African haplogroup, the L haplogroup. In fact, there's only 6 patients belong to the L haplogroup in this 420 Ewing sarcoma patient cohort. If you just look at the self-reported risk and the ethnicity, and this is kind of, again, as we have always been arguing, and kind of actually in the messy, right? We don't really understand the pattern. Even if you group the patients by their mitochondrial macrohaplogroup, LM and NR, you see that in patients that belong to the L haplogroup, the African ancestry haplogroup, you see there are still patients reported to be in the white and not Hispanic and the Latino. And similarly, in the R microhaplogroup, you have patients reported with all different race and ethnicity. So it's not that easy to sort out what is the contributing factor for the increased risk for Ewing sarcoma. So what we decide is to use, of course, computation determined genetic ancestry. And by this entry, we use 130,000 so-called ancestry informative markers and determine the local ancestry of this patient nuclear ancestry of all these Ewing sarcoma patients. And results are shown here, right? We can see that based on the genetic ancestry, nuclear ancestry of these patients can be grouped largely into 6 different groups, with the largest group, the group in the middle, with the primary ancestry from Northern European, Mediterranean, and Southwest Asian. But then you could also see that there are patients with primarily Northeast Asian ancestry and in light blue, and also the Native American ancestry in light blue, darker blue on the right-hand side. But still, if you look at it very closely, you see that it doesn't matter which group this patient fall into, they all have pretty significant percentage of European ancestry. That is very interesting to us. Now we're trying to put the two things together, and we know that there's a strong bias toward actually mitochondrial ancestry of European origin, and there's strong bias against mitochondrial ancestry of African origin. But at the same time, there's strong actually enrichment of nuclear ancestry in with European origin as well. When we try to put this together in this plot, you see that there are some patterns start emerging, right? On the right-hand side, you see that with the pie chart, there's indeed indicate that there's significant enrichment of the European microhaplogroup R. 90% of the patients belong to this microhaplogroup, and only 1.4%. In fact, there's significant impoverished of patients belong to the African L haplogroup. And on the left-hand side, you say if you group the patients by the LMNR, the mitochondrial microhaplogroup, you see that even in the patients belong to the L haplogroup, there's a significant percentage of nuclear ancestry from some of the ancestral group that belong to Europe, right? For example, and the Mediterranean and the Northern Europe and Southwest Asian populations as well. Now, this is actually a very busy plot, and try to apologize and in advance. And so this is us trying to look at each individual sample one by one and shown in each line, right? And this is 5 panels of 84 patients each, and each patient, the gene ancestry is depicted in one line shown here. And this is the horizontal line indicates the percentage of local ancestry from each of the 9 ancestral group, and followed by the mitochondrial haplogroup designation, and whether it's from LM and NLR, and followed by the self-reported race and ethnicity. We're trying to point out that you cannot really totally rely upon, for example, the self-reported race and ethnicity, as I try to point out with some of the arrows on the right-hand side. You see that even though most of the patients indeed have almost exclusively European ancestry in terms of both nuclear and mitochondrial ancestry, there are few individuals here and with self-reported, for example, African Indian, American Indian, or Alaska Native race, and some other actually and ethnicity as well. What's interesting is the group of 84 patients shown on the leftmost panel, right? You see there are a number of patients that point out by the red arrows. There are patients belong to the L haplogroup, 6 patients. And in fact, 4 of the patients still have primarily European ancestry in their nuclear ancestry analysis. And only 2 of the patients have primarily African ancestry. And even for these 2 patients, there are significant percentage of nuclear ancestry. And there are actually a number of patients here shown in the middle, point out by these 2 arrows. They are the individuals with almost exclusively, for example, Native American nuclear ancestry, also exclusively Asian ancestry in the nuclear ancestry, but they do belong to the largely Eurasian microhaplogroup R. So these are the probably a few exceptions. In the one, just want to actually outline the few conclusions that we have. First of all, we believe our study really established the landscape of mitochondrial DNA mutations in pediatric cancers. We showed that the deleterious mitochondrial DNA mutations contribute significantly to the pediatric cancer tumorigenesis, and we found a number of hotspots for mitochondrial loss of function mutations we believe that they point to certain mitochondrial genes and pathways and suggest they may play a bigger role in tumorigenesis than others, especially complex 1 genes and ND4 and ND5. And we believe that the different mitochondrial DNA mutations are associated with different pediatric cancer subtypes and contribute different tumorigenesis, tumor progression of these tumors, including pediatric CNS tumors. And we believe that the mitochondrial ancestry may play a bigger role in the increased risk for some of the cancer types, such as Ewing sarcoma. In the end, just want to acknowledge all the people have made significant contributions to each of the 3 studies that we just published the last few years. And thank you.
This is maybe more a broad question about mitochondrial biology and cancer, but when I think about cancer growth and tumor growth, I imagine these very aggressive, cell growth, very rapid, lots of energy is involved. So how do these loss of function mutations in mitochondria, it feels to me that might make the mitochondria less powerful to fuel these kind of growth. Could you talk a little bit about that interaction?
Yeah, absolutely. And I for the longest time stuck with the idea that the mitochondria is a powerhouse, right, and of the cell, and that's what we commonly say. I think I was misled by that as well, and forgot the other aspect of mitochondrial biology. In fact, mitochondria play a much more important role than just being a powerhouse, and there's a lot of things like signaling and so on and so forth. There's one aspect of mitochondria that's underappreciated. That's my working hypothesis. Right now is that these mutations, when they accumulate in the mitochondria, they actually in the cells, they trigger the cascade of pathways, including like the mitochondria unfolded protein response pathway. All these pathways are known to infect and to exert the roles beyond just energy production, right? And they are important for all aspects of cellular functions, including actually tumorigenesis and so on and so forth. So I think when we keep on thinking of mitochondria as only as a powerhouse, and we are misleading ourselves, and that's my current belief. And so this loss of function mutations, they don't only affect the energy production, they affect other things as well, and just not being studied enough. And so I'm a big advocate for looking at this from all different perspectives.
Dr. Heath is the Director of Data Technology and Innovation at the Center for Data-Driven Discovery in Biomedicine at the Children's Hospital of Philadelphia. So she's one of the PIs of the Kids First Data Resource Center. And so she's going to be talking a little bit about how Kids First data can be interoperably used in other large research efforts. So with that, I'll pass the screen to Dr. Heath.
Great, thank you, David. And it's great to go after 3 amazing talks, I think with a lot of cross connection, and really enjoyed the discussion. So with that, I'll try to be a bit timely because I think there's a lot of good questions that are popping up, and hopefully part of what I'll do is try to motivate some more of that we've heard a lot about folks, I think, already using Kids First data in conjunction with other data sets, trying to integrate them. And I think this is something we recognize very early on with the Kids First Data Resource Center, that while we are a hub of the pediatric data, really the power becomes when you can integrate it with other data sets, ask new hypotheses, make sure that the data is interoperable in ways where you can immediately get to these kind of very interesting questions that are popping up. So I'll recall a previous slide that, we really are part of the larger data ecosystem. I mean, and anyone doing data these days is part of a larger data ecosystem. And, right in late 2019, this idea of the NIH cloud-based platforms interoperability got stood up. We were one of the initial partners there, and really started to think and talk through the fact that we have our different data domains, but especially on the genomics component, being able to bring those together. We've already had some questions on how does that work? How hard is it? And really with this idea that, we need to be able to streamline that and make that easier as more of this data is generated, more of the data is released and made available to the public. And so, we have been really trying to think through what are the right ways to do this? How do we make sure Kids First data is always accessible? How do we make sure we're prioritizing use cases that are most impactful to our community? And what are all the different technical layers that underpin that? So I'm going to talk through two kind of use cases that we've worked through, and then probably end a little bit on, what are everyone else's use cases, especially as we continue to move forward. We've kind of, through some of these initial use cases, created a foundational level of technology that allows us to really get to the interesting science. So the first one was a use case that was formulated a couple of years ago around Kids First and the undiagnosed disease network data. So for anybody who's worked across these, UDN is also doing a fair bit of sequencing. There's a lot of pediatric cases in there, and there were a lot of scientific use cases that came up around this. And they have actually most recently released their data on dbGaP. They now have a little under 4,000 whole genomes available, a little over 4,000 genotypes, RNA-seq, whole exome as well. And all this data resides in dbGaP and SRA. And so this was a kind of key use case to ask, what would it really take to be able to integrate Kids First data with data that lives in dbGaP and SRA? How does that work with the genomics? How does that work with the phenotypic access? How does that work with the fact that these are controlled access? And really starting to work through those key pieces. And so, the idea being that kind of looking around the different standards, I think two have emerged for the Kids First ecosystem over the last couple of years, and increasingly you'll see it's being widespread across others. So the GA4GH DRS standard is really the data repository services that allow us to index controlled access data, provide the equivalent mechanisms to feed that into pipelines, and really start being able to, choose your cloud workspace of choice. For Kids First, we're obviously integrated with Kavatica pretty directly, so that's the first place. But the idea is that, that's just a first step, that any cloud platform of interest, really using these standards, you should be able to port this around. And then the other standard that's really come up in the Kids First ecosystem is around FHIR, F-H-I-R, the HL7 standard, which is increasingly becoming a flexible API that allows us to interchange different data. And so it became this driving use case of what would it really take across these two resources to have some of the clinical phenotypic data in FHIR, the genomic data files indexed in DIRS, and to be able to bring them into a workspace and co-analyze it. So since the very beginning of Kids First, we've always had this button of Analyze in Kavatica that allows you to search and bring your data over into that. Over the years, that's gotten better. There's been more metadata created, and it's moved from a custom solution when we first started to now when you do this, you'll see this little DRS bucket icon there and then highlighted in there the actual project study name. And that is using the standard, the GA4GH standard that I talked about earlier. So we've moved from having a custom solution to doing this to now using a standard that anyone can use, including to going to places like Terra or other cloud platforms that support DRS. So really this became working very closely with the dbGaP and SRA team to understand what would it take to allow the UDN data that had been deposited there to be also immediately available in this way. And right now we have this solution, although we are continuing to work with dbGaP and SRA of how do we generalize this, of having brought and indexed their DRS services, so using the same standard with dbGaP, index them in Kavatica and allow you to bring those files into the same workspace that you had your Kids First data. So now if you have access across both UDN and Kids First, you will be able to bring those into the same workspace. Now, if we're doing our job right, this also means that those DIRS URLs are available to anyone once again, but this is kind of what it looks like from a Kavatica perspective of going in there, finding the different genomes of interest and being able to bring them into the workspace to analyze. And this is powered both by the standards, but also an important piece is how do we do the controlled access? And really this has been enabled by the NIH researcher auth service. So if you've logged into places over the last year or two, you'll notice that the ERA logins have changed, and you'll see this kind of screenshot in the lower left here that prompts you to log in via RAS. And so it's really been moving the former ERA commons into a more modern authentication stack that allows us to have these integrations such that when you log into Kavatica, it can relay those RAS passports both to the Kids First DRS service as well as to dbGaP, as well as to other RAS integrated services, and vice versa. If your cloud workspace of choice is a different place and it's RAS enabled, it can also call it to these same places and bring it in. And so it becomes this way that we can really start thinking about these, integrated yet controlled access and protected ways of accessing the controlled access data in a more seamless way. And so at the end of the day, what it, actually looks like that you can go do right now is bring in the different data sets into the project, have your analysis. Obviously, this is a really quick SAMS tool, index and be run, but you can run the different Jupyter notebooks, RStudio, all the other features of Kavatica can leverage and use these files as well. And as I said, it's becoming more widespread. So, we have this nice kind of integration between our portal and Kavatica. Maybe we don't have that integrated for every single cloud workspace, but more and more what we're being able to do is export some sort of DRS standardized manifest, bring that to a cloud platform of choice, and bring that data in. So in Kavatica, this is what it looks like. You can bring a CSV file from a different place that might not have a direct integration, such as Kids First or UDN. For example, the Cancer Genomics Cloud or other places, Biodata Catalyst, Anvil, et cetera, and be able to bring that into your project and be able to co-analyze it. And so, yes, you might not have the nice portal interface, but again, like I'm saying, this underlying technical infrastructure allows us to do these things where you can go and find the different data sets of interest, UDN, Anvil, Kids First, CRDC, Biodata Catalyst, and start bringing them all into one unified project to be able to analyze and work with. And so, it was really great to hear some of the different use cases across wanting to co-analyze top med data and Kids First data and other things like that are important. And so this becomes reusable. So while we are showing this in Kavatica, there's no reason that any other cloud platform that wants to implement these standards can do this as well. And so this is what this last one's saying, that yes, I'm showing the kind of the data coming to Kavatica, but similarly, we want to make the Kids First data widespread because we recognize that no one platform is going to build all tools for all things. There's different focus on different aspects. There's different platforms that are better for different types of analysis work. And really, you should be able to move through those and use the best of breed for whatever work that you are doing. So I'll wrap up the UDN use case. I have one more on include. I do want to give a shout out. I think David had acknowledgement across all the Kids First DRC, the technical teams who've really been hands-on doing this, especially with the NCPI, as well as the Common Fund Data Ecosystem under, UDN has been a Common Fund data set as well, which has really helped enable this work. And a big shout out to Michele Mattioni at Belsera, formerly Seven Bridges, who's really spearheaded a lot of this work. I'm just really speaking on behalf of these different technical teams. So I'm going to talk a little bit about Kids First and Include, and then I'll wrap it up. And really, this has been an interesting synergetic kind of partnership, I guess, across different Down syndrome genomics, and really trying to think through what does it mean to start having these federated systems that aren't just what I showed you with Kavatica, but rather how do we have portals? How do we find the data to make it searchable, findable? How do we have different communities and projects where Kids First, as you heard, focus on a lot of the developmental biology aspects, a lot of the kind of broader pediatric genomics, while Include really focused on Down syndrome. How does the different aspects happening with chromosome 21 impact how we want to harmonize data, how we want to provide that, how people curate it, and really thinking across, how do we federate these things? And so when Include launched, Kids First had actually helped through the XO1 mechanism fund initial cohorts of Down syndrome. And so while those live in the Kids First Data Resource Center, we really want to expose them and make them more searchable and include. And so you can see a snapshot here of the different studies where the ones that are include/Kids First are really being indexed, brought in, and searchable in the include portal in ways that maybe more focused for the Down syndrome community versus if you came through Kids First, where it's more of a broader landscape. And so what that allows you to do is that because we have both done a lot of the clinical data curation with Mondo and HPO, as well as the genomic data harmonization, you can immediately go through and start searching across these different studies. The studies that are kind of coming through include are curated similarly, and you can immediately start searching and looking for the different data sets of interest. And so you might ask kind of, how's this done behind the scenes, or you might not, depending on who you are. But really what we have kind of thought through is some of this aspects of setting up FHIR APIs to be able to interchange this data. And so what's important here is that FHIR allows a standardized API where we can put elements that if they're curated the same, for example, to HPO and Mondo, we can agree and put them in the same places. But if we have other data elements, for example, on include, we have a very specific Down syndrome status, which on the Kids First side, we're not going to curate. And on the Kids First side, we've done a fair bit of work on, for example, some of the cancer aspects that, include will, be focused on some of the leukemias and other aspects of the comorbidities that happen between the two. And so we can provide these interoperable APIs, and then the different portals and the different front-end features can pick and choose which ones do I really want to bring in and index and make available to my community, and make it available in different ways. Kids First is very genomics focused. Include has a lot of genomics, but there's also proteomics, metabolomics, other molecular assays, as well as imaging coming online. And so it allows us this infrastructure where when it's the same, let's go ahead and index it. If it's not, that's fine. We might have different domain specific things that are happening across there. And so what's really happening in the front end here is you're seeing the include data from the include FHIR API being indexed in the portal, and then a slice of the Kids First data that's relevant to the include community being cross-indexed. And then as include data becomes more and more available, we anticipate being able to do the same in Kids First. So as pediatric cohorts or other things happen and include that make sense to pull into the Kids First portal, we have that infrastructure and that framework to start doing the same. And if you're really interested in the super techie, weedy details, there's a couple endpoints there to look at the actual FHIR services, except I copy and pasted the include one wrong. So I'll update that. And then the last part is it's not just our portals and our development teams. What we're really starting to see is that this becomes an interoperability framework where others can build tools. So for example, NLM has a research data finder tool that you can put a FHIR API in and start looking at different FHIR APIs. So the dbGaP actually has a FHIR API that you can put this in. We're starting to see examples in libraries being built around it for Jupyter or RStudio, if you want to bring it into pipelines, where it becomes a substrate for it's not just the portal application developers, but really any kind of tool or application development here. And then you have similar abilities to pull different data into the application tools of interest. So I also think that the include team, especially the include DCC data hub team, some of the different also technical folks, especially Jeremy and JP at St. Justine, who have done a lot of the work on the portals, Vanderbilt, Robert Carroll, and Eric Torstensen, who've really thought a lot about the FHIR mapping, and Mean on the CHOP team, who has really done a lot of the legwork for the development, and our partners there, the Linda Stoner Institute, SAGE, Valsera, and the TIS lab at Colorado as well. So with that, I think I'll really just end on this concluding slide, and I hope to hear both discussion on the scientific side, interoperability. We're definitely around. We're really thinking through this. So it's really, as people have different use cases here, thinking through all the different, ecosystems. We've been involved in CPI, we've been involved in CFDE, and then most recently the CCDI Federation recently launched. So it was really nice to see some of the co-analysis data with the St. Jude Cloud as part of the sarcoma work. The other partners there, the Pediatric Cancer Data Commons at UChicago and the Treehouse Childhood Cancer Initiative at UC Santa Cruz. And we are, as that's just launched, thinking through use cases. How do we do this on the pediatric cancer side? We've seen some of the rare disease with UDN. We see some of the syndromic work with Include. And so really just thinking across all these different scientific aspects and finding the common framework that we can use across all these different use cases as we move forward. So with that, I think I'll turn it over to questions and or discussions for my part or any of the previous speakers.
Thank you, Dr. Guy and Allison, for sharing your experience with secondary use data and also interoperability. So my question is more technical again, like which challenges should researchers keep in mind in terms of how to move to use for the secondary use of the data and interoperability projects, what challenges researchers should keep in mind when they're planning their studies, etc. Can you comment on that in vision of your own research? Thank you.
Yeah, so from a technical standpoint, I'll say we've made a lot of progress over the last couple of years, but it's still not point and click. I definitely think that you still need some technical skills right now to navigate it. I think it's partly why it's important to think critically about which data sets do you want to pull in, understand, I think the questions you're trying to ask, is it a scientific question? Are you trying to develop methods? Which data sets are going to be the most important to spend your time and effort on obtaining and making sure will help what you're doing. I think the other aspects what other people alluded to is there's always some level of harmonization that needs to happen. We try our best at Kids First to make our data sets harmonized, but we have been around for 5 years, and I think as someone alluded to earlier, some of the earlier data sets were previous pipelines, and we're actively discussing what it would mean to bring everything up to the most recent. And the different trade-offs there. I do think that's a place where some of the cloud credit aspects can be helpful to be able to fund that kind of last mile harmonization, whether that's calls or other aspects. I will say that the other thing is really thinking about what kind of phenotypic and clinical data is really of utility, that there's high variability in what's provided across different studies. Again, we try to curate it to things like HPO and Mondo, but a lot of times, you find something very interesting genomically, and then, the next question is what's happening clinically, what's the outcomes, and I think that's generally a challenge for the overall community. I think we are seeing the trends of that getting better and easier, and it's part of why we're also keen at the interest in FIRE, because it provides a pathway to more longer term EHR follow-up or other things that might be able to kind of lower some of the barriers to understand what's happening longitudinally. So I'm not sure, Dr. Guy, if you have some response to that as well.
I feel like Allison, you're not giving you guys enough credit, right? I think you're building such a tremendous resource, and what I would say that they really level the playground, right? And to a lot of researchers, without a lot of resources, access to a lot of people, right? And so, they just have some maybe smart ideas, and this really enable them to ask some very interesting questions, very smart questions, without having to have that tremendous amount of resources and so on, especially for the young investigators, right? And I know that when you just first start your career, you don't have a overall for a few hundred Ewing sarcoma patients, right? You don't have the amount of funding to allow you to do the whole genome sequencing of that many patients yet. If you have a particularly interesting question, you can ask. In fact, from my own experience being a bioinformatician, I feel like it's extremely useful already, right? And I can just gain access to the data and from command line over the web, and using just mount that as a remote share drive, and it's quite easy to do some analysis. Also, the pipeline that you're using are top tier and high quality pipelines. I know that I didn't even have to call the variants myself, right? And I know you're running, for example, the Dragon pipeline. About calling the variants. These are essentially the pipelines that will also validate in our lab. We're clear certified diagnostic lab. In fact, we use that for our whole genome sequencing and OSN sequencing test, the Dragon pipelines. And so I have a lot of confidence in which you guys have achieved and accomplished.
And Dr. Guy, if I can follow up, how did you find the data at the Kids First portal?
So I guess personally, I look for data, right? And look for a large data set. I like to pursue a kind of data-driven research, not necessarily all hypothesis-driven. And just, I'm a firm believer that you should be letting data tell you what's interesting, and instead of the other way around. So the fact that I know that from my colleague, in fact, I mentioned at the very beginning, I used to work at CHOP. So of course, heard from my former colleagues that there's a tremendous resource being established. Of course, I'm very interested in exploring and finding out. In fact, aside from the 2 studies I just presented, we are also pursuing a number of other studies trying to take advantage of the Kids First data sets. We hope that we'll turn them into publications real soon as well.
So I think there's time for one more. So I thought Dr. Seidman was asking about some of the mechanism between mitochondrial ancestry and nuclear genome ancestry. And so I might let him explain a little further.
So it sounded like there's a discordance between the nuclear ancestry and the mitochondrial ancestry, but that, to me, it seems like that should be expected, right? Because mitochondrial inheritance is 100% maternal. So if the father is, let's say, of one ancestry and the mother is of another ancestry, it's only going to look like the mother's ancestry is inherited in the mitochondria. So I'm a little, I don't understand why we'd expect so we should expect to see discordance, I would say, between nuclear and mitochondrial ancestry. But maybe I'm not thinking about it.
No, I agree with you 100% there. And you expect to see some discordance, indeed. And what I'm trying to, the point I'm trying to make is just by integrated analysis of nuclear ancestry and the mitochondrial ancestry, we will have a better understanding of what is a contribution of genetic background to the increased risk for Ewing sarcoma, right? Instead of just looking at the race, ethnicity, and as a categorical number values, right? And so it being white or being actually Hispanic, being actually in the end, and so we look at the mitochondrial ancestry is kind of interesting, atomic. I call it atomic, right? And either this haplogroup, that haplogroup, but then you have a nuclear ancestry representing kind of a continuous value, right? And with different percentage of genetic ancestry from all the different ancestral populations. It's kind of very interesting way to think the question. And I'm proposing that maybe the best way to understand the contribution of genetic background is to combine these two together, right? And looking at from that angle instead of exclusively only using this versus other. In fact, no.
Gotcha.
I just want to thank you, our speakers, and make a comment. Like you four have such different research topics, but you all shared today how Kids First was kind of a central piece for your research. So thank you for giving these examples to the research community and being here with us today. And David, I'll give you the final word.
Yeah, that's about what I was going to say. But thank you so much to all 3 of our speakers, as well as Allison, for joining in and sharing this great research that's going on with Kids First data. We're so happy to see it. And so with that, I'll close the webinar here. Thank you so much, everybody, for joining. And we hope to see you at future Kids First events in the future.